everyone, and welcome to our module on myelopoiesis. Leukopoiesis is the formation of white blood cells, and in terms of the divisions of leukopoiesis, we have it being divided into two major forms. We have what we call myelopoiesis, which we talk about in this module, and lymphopoiesis, which we talk about in a separate module. And myelopoiesis is the formation of specifically granulocytes and monocytes. Granulocytes includes the likes of the basophils, the eosinophils, and the neutrophils, and then monocytes are a special categories of white blood cells that usually become macrophages once they enter into tissues. This is an overview of the process of myelopoiesis. Myelopoiesis, just like any other form of hemopoiesis, starts from the stem cells in the bone marrow. Stem cells eventually become what we call progenitor cells. In the case of the process of myelopoiesis, the stem cells eventually become myeloid stem cell, and the myeloid stem cell eventually would become the progenitor cells for the granulocyte and monocyte lineage. Then from there, these progenitor cells would differentiate and would form what we call precursor cells. So we have to start with the stem cells. Stem cells become progenitor cells, and then progenitor cells becomes the precursor cells. And we have different forms of precursor cells at different stages of maturation that would eventually give rise to those granulocytes and monocytes that we talked about. So we'll talk about each of these at different stages. We'll start with the stem cell stage, then we'll talk about the progenitor cell stage, and then we'll focus on the precursor cell stage at last. Granulocytes and monocytes are derived from a special form of stem cell in the bone marrow known as pluripotent hematopoietic stem cell. These are stem cells that have the ability to self-renew themselves and also self-differentiate, but are not morphologically identifiable. The granulocytes and monocytes derived from these pluripotent stem cells tend to be specifically originating from a form of stem cell that is differentiated from the pluripotent stem cell. And this is what we call the myeloid or the GEMM committed stem cell. So the pluripotent hematopoietic stem cells differentiate into two forms, differentiate into the myeloid stem cell or the lymphoid stem cell. And it's from this myeloid stem cell that we have the granulocytes and monocytes being derived from eventually. So the myeloid stem cell or the GEMM stem cells tend to differentiate into three major progenitor cells. This would include the post-forming unit erythroid, which tends to go ahead to form erythrocytes through the process of erythropoiesis. We talked about that in a separate model. Then the post-forming unit megakaryocytes, which also undergoes thrombopoiesis to form thrombocytes or platelets. And then finally, the highly proliferative potential colony forming unit, or HPP CFC for short, which we'll be talking about in the next slide. This is the major progenitor cell for the process of myelopoiesis. Now moving on to the progenitor cell stage, we start with the highly proliferative potential colony forming cells, also known as HPP CFC type 1 cells. This is the first progenitor cell under the lineage of myelopoiesis, and what you need to remember about the progenitor cells is the fact that they are cells that are committed to a particular cell lineage. They can undergo cell division and cell differentiation, but they can't undergo self-renewal, and that's in contrast to the stem cells, and also they are not morphologically identifiable, and that's in contrast with the precursor cells that we're talking about next. For the type 1 cells, undergo cell division and differentiation to form the highly proliferative potential colony forming cells type 2, which we'll talk about in the next slide. Now, the highly proliferative potential colony forming cells type 2, the second progenitor cell stage, they further divide into subtypes of progenitor cells, which we'll talk about next. This includes the granulocyte macrophage colony forming cells, the eosinophil colony forming cells, and the basophil colony forming cells. Then for these progenitor cells to form precursor cells, they would also need to undergo cell division and differentiation. What we need to know is that, first of all, the granulocyte macrophage colony forming cells first divide into subunits of progenitor cells. they divide into the granulocyte colony forming cells and the monocyte forming cells. And then from there, each of these progenitor cells would eventually give rise to precursor cells that would continue the process of myelopoiesis. So we have the granulocyte colony forming cells, the eosinophil colony forming cells, and the basophil colony forming cells, giving rise to a similar type of precursor cell called the myeloblast. And then finally, the monocyte colony forming cells then to form its own separate form or separate type of uh, precursor cell known as the monoblast. Now, from here, we talk about the process of myelopoiesis in two different um, forms. All right, depending on whether we're talking about the myeloblast or the monoblast. In terms of the myeloblast tend to undergo the process of granulopoiesis to give rise to granulocytes. If you pay attention, you notice that the myeloblast is gotten from progenitor cells that are specific to either granulocytes, eosinophils, or basophils. So the myeloblast that is formed from these different progenitor cells goes ahead to undergo what we call granulopoiesis to give rise to granulocytes. Then the monoblast gotten from monocyte colony forming cells, which is a progenitor cell, will also go ahead to form monocytes and macrophages through a different process of myelopoiesis, which is called monopoiesis. So we'll talk about each of these two different forms of myelopoiesis: granulopoiesis, which entails the further maturation of the myeloblast, and monopoiesis, which entails the further maturation of the monoblast in the next couple of slides. Let's start with granulopoiesis. Granulopoiesis begins with the first precursor cell under its lineage. That precursor cell is the myeloblast. The myeloblast is a mononuclear cell, and what is unique about its nucleus is the fact that it has a very large nucleus that tends to take about 90 to 98% of the entire space. And you notice that the amount of cytoplasm or the volume of cytoplasm is very minute in contrast to the nucleus. The nucleus is ovoid in shape and has in terms of its cytoplasm, it has very few or no primary cytoplasmic granules present within it. We'll talk about what cytoplasmic granules are when we get to the next other the other forms of precursor cells under granulopoiesis. Myeloblasts tend to eventually undergo cell division and maturation to give rise to the next stage of precursor cell under granulopoiesis known as the promyelocyte. Promyelocytes are the second precursor cells under granulopoiesis. They are smaller in size compared to the myeloblasts, as you can see. They also contain a they are also mononuclear cells that contain a single nucleus that is ovoid in shape, but this nucleus is slightly smaller compared to the myeloblast. When we talk about the nucleus of the promyelocyte, it's important to note that they have a prominent nucleoli. The nucleoli gets less prominent as we go down the different precursor cell stages. So the most prominent nucleoli is found within the myeloblast. Then from there we have it being less prominent in the promyelocyte, and then finally in the myelocyte, which is the third stage of granulopoiesis. One important thing to note about the promyelocyte is the presence of primary cytoplasmic granules. Now we saw either very few amounts or even no primary cytoplasmic granules when viewing the myeloblast, but in the case of the promyelocyte, it is said to be to contain a rich amount of primary cytoplasmic granules. What are these primary cytoplasmic granules? So these granules are rich in a number of enzymes, usually what we call myeloperoxidase enzymes, and these enzymes are there for the function of killing microbial agents that have been phagocytosized by the granulocytes. So when we have these cells eventually maturing to form the granulocyte that we talked about at a certain stage, one of the functions of the granulocyte is to undergo phagocytosis, and how the microorganisms that gets phagocytosed into the granulocytes gets destroyed is through the presence of this cytoplasmic granules. So the enzymes within them are important for breaking and destroying the microbes that have been phagocytosed by the granulocytes. So the very first cell, precursor cell that we tend to see large amount of primary cytoplasmic granules tend to be the promyelocyte, and this is the hallmark in recognizing it. The promyelocytes undergo cell division and differentiation to give rise to the third stage of precursor cell stage of granulopoiesis known as the myelocytes. So myelocytes are the third precursor cell stage of granulopoiesis. They are smaller in size compared to the promyelocytes. They are also mononuclear cells containing an ovoid-shaped nucleus. And one important contrast is that the myelocytes tend to have a combination of both a primary cytoplasmic granule and new set of cytoplasmic granules we call secondary cytoplasmic granules, however few. Also, another point of difference to note between the myelocyte and the pro-myelocyte is the difference in prominence of their nucleoli. The nuclei of the myelocyte tend to be less prominent and less visible compared to that of the promyelocyte. Let's pay attention to the secondary granules. So, what are secondary granules in contrast to primary granules? Secondary granules, unlike the primary granules, are usually large in number but smaller in size. Um, the light microscopy is incapable of visually resolving the size of the secondary granules. And then we usually see that they appear like sands deposits in the cytoplasm of the myelocytes. And usually the content of the secondary granules tend to differ and helps in differentiating the myelocyte into three different forms. So we could have a basophilic myelocyte that contains special forms of secondary granules that distinguishes it from an eosinophilic myelocyte, which is also distinguished from the neutrophilic myelocyte. So different forms of secondary granules would help in differentiating myelocyte into either a basophil, eosinophil, or neutrophilic myelocyte. But in terms of the myelocyte, remember that it contains both primary and secondary granules being fewer compared to the primary granules. The next stage of the development of granulopoiesis is the metamyelocyte. Metamyelocytes are gotten from myelocytes after they have undergone cell division differentiation. They are the fourth precursor cell in granulopoiesis. And one unique thing about the metamyelocyte is the process of indentation that occurs to the nuclei. Right before now, we've been talking about the fact that from the myeloblast all the way down to the all the way down to the myelocyte, the nucleus of each of those cells tend to be ovoid in shape. When we get to the metamyelocyte, what changes is that that ovoid shaped nucleus becomes indented and gives rise to what we call a bean-shaped nucleus. So the bean-shaped nucleus is the hallmark of the metamyelocyte. Metamyelocyte also lacks a nucleoli. So remember we talked about the prominence of the nuclei decreasing as we go from the myeloblast down to the myelocyte. From at the metamyelocyte stage, we no longer can see or visualize the nucleoli. And finally, the metamyelocyte contains large amount of secondary cytoplasmic granules. The primary granules actually being replaced by secondary granules at the metamyelocyte stage. And these secondary granules, as I said before, also helps in distinguishing the different forms of metamyelocytes into either basophilic, eosinophilic, or neutrophilic metamyelocytes. Metamyelocyte undergoes cell division, and differentiation to give rise to the next stage of granulopoiesis, what we call the band cells. The band cells are the fifth precursor cell for under the process of granulopoiesis. They are cells that are larger in size compared to the metamyelocytes. They undergo further nuclear indentation. By nuclear indentation begins at the metamyelocyte stage. This process continues all the way up to the band cells, and it gives rise to cells that becomes bilobed or multilobed in terms of the amount of lobes of the or indentation that is found or formed in the nuclei. As you can see in the image below, you notice that the indentation found in this particular set of band cell makes or deepens the nucleus, making it to be bilobed. So it has one lobe here and then another lobe here. Some cells could have multiple lobes compared to uh, others. Some have about three, some four, some five, and some just two lobes. Another very important thing about the band cells is the presence of secondary cytoplasmic granules. This is also found in the metamyelocytes. The amount tends to be almost equal to that of the metamyelocytes, and the nature of the secondary granules would differentiate or distinguish different forms of band cells into either basophilic cells, eosinophilic cells, or neutrophilic cells. We'll talk about each of the mature cells that the band cells tend to give rise to: basophils, eosinophils, and neutrophils, based on the nature of the secondary cytoplasmic granules that are formed. Moving on to monopoiesis, which is the second form of myelopoiesis. Monopoiesis begins with the first precursor cell under its lineage, which is the monoblast. Monoblast is a mononuclear cell that lacks cytoplasmic granules. As you can see in the image, there are no cytoplasmic granules present within the cytoplasm of a monoblast. It is similar to that of the myeloblast in the sense that the nucleus tends to occupy a large chunk of the entire cell volume. Other things to note is the presence of prominent nucleoli and presence of vacuoles within the cytoplasm. Monoblasts would eventually divide and differentiate to give rise to the next precursor cell stage under monopoiesis, known as the promonocyte. So promonocyte is the second precursor cell stage. It is also a mononuclear cell, but compared to the monoblast, the nucleus of the promonocyte tends to begin to undergo indentation. The indentation of the promonocytes tend to come with immature chromatin present within it. Vacuoles are also seen within the cytoplasm, but we don't see cytoplasmic granules as compared to that of the cells under granulopoiesis. A promonocyte undergoes cell division and eventually cell differentiation to give rise to the monocytes. The monocytes are the third precursor cells under monopoiesis. They are mononuclear cells with a nucleus that is indented, or what we call kidney shaped, as shown in the image. So the nucleus of the monocytes is very synonymous or very similar to the nucleus that we see in the band cells or the metamyelocytes. Remember that each of them have indented nuclei. So it's the same thing that we see in the monocytes, but we usually just describe the nucleus as being a kidney shaped nucleus. Monocyte also lacks cytoplasm, and one important thing to note about the monocyte is the fact that once it is formed within the bone marrow, it begins to migrate into the bloodstream. Once it gets into the bloodstream, it resides in the bloodstream for about 8 days, of which it finally leaves the bloodstream and enters into the tissues. The process or the stage at which it enters into these tissues, we stop calling it the monocyte and we start to refer to the monocyte as a macrophage. So at the end of myelopoiesis, whether granulopoiesis or monopoiesis, we would give rise to four different forms of cells. This includes the neutrophils, the eosinophils, basophils, which are gotten from granulopoiesis, and then the macrophages or monocytes, depending on whether they are within the tissues or within the bloodstream, respectively. And those are gotten through monopoiesis. Now let's talk about each of these different mature cells and what makes them unique, starting with the neutrophils. Neutrophils are mature granulocytes that contain a multi-lobed form of nucleus. Remember that during granulopoiesis, the band cells tend to undergo further indentation to give rise to a nucleus that is multi-lobed or bilobed. In the case of the neutrophils, the nucleus tends to be multi-lobed, as shown in the image here. And another unique thing about the neutrophil is the presence of a specific form of secondary cytoplasmic granules. Now, the neutrophils contain a secondary cytoplasmic granule that is both acidic and basic in nature, contains both acidic and basic macromolecules. And this is important when we stain. So, when we stain to view a neutrophil under the microscope, as shown in the image here, what happens is that because it contains granules with acidic and basic macromolecules, these granules attract both basic and acidic stain, and that makes the cytoplasm to appear a bit pinkish rather than either reddish or bluish. So it has a mixture of both basic and acidic stain or dye, and the neutrophils appear pinkish or neutral, hence the name neutrophil under the microscope. The second granulocyte is the eosinophil. Eosinophils are um, granulocytes that tend to have a bilobed form of nucleus, And within their cytoplasm, they have granules that stain particularly with eosin. The the granules present within the eosinophils tend to be rich in basic or um, alkaline materials, alkaline macromolecules, and as a result, these alkaline or basic granules tend to attract a lot of acidic dye, and that attraction of acidic dye is what gives the eosinophils a reddish appearance when we stain under the microscope. We tend to see a lot of eosinophils come into play whenever we have a parasitic infestations, whenever there's an intestinal worm in the system, or um, or maybe a metazoan or a helmet infection. Usually, we see eosinophils tend to rise, as some of the granules we have within the eosinophils are specifically involved in lysine cells or cells, the cell surfaces of these parasites. The third granulocytes are the basophils. Basophils are um, granulocytes that have also bilobed nucleus. Particularly, they have um, cytoplasmic granules that are rich in acidic components, in acidic macromolecules. And as a result, when we stain the basophils and view them under the light microscopy, they tend to appear bluish because of the acidic content of the granules tend to attract basic stain. Basic stain is what gives the basophils this bluish uh, coloration under the microscope. Basophils are seen in inflammatory or hypersensitivity reactions, like allergic reactions. So whenever there's an asthma, for example, or atopic dermatitis, the culprit cell involved in this process usually would be the basophils. Basophils within the bloodstream are regarded as basophils whenever they leave the bloodstream to enter into the tissues, as seen in asthma and other allergic conditions. We refer to them as mast cells. And usually, remember that mast cells tend to degranulate through an inflammatory reaction to give rise to different forms of systemic or local effects. And that degranulation is just a release of the secondary cytoplasmic granules that we see within the basophil. So this is a picture of a mature monocyte. Remember that monocytes are referred to as monocytes when they are found within the bloodstream. Whenever they leave the bloodstream and enter into the tissues, we refer to them as macrophages. Well, monocytes, remember, contains no cytoplasmic granules. As you can see, the cytoplasm is virtually empty. One unique thing about them is the presence of this large kidney-shaped nucleus. One other thing to note about the monocytes in terms of once they leave or cross with the bloodstream to enter into tissue is that the macrophages that they tend to form tend to be phagocytic, and this mirrors what we see in the neutrophils. Neutrophils also are phagocytic in nature. They tend to undergo phagocytosis to destroy bacterial organisms. Macrophages, on the other hand, tend to undergo phagocytosis and are referred to at different names with different names at different tissues. So, for example, macrophages in the brain is referred to as a microglia cell. Macrophages in the liver is known as a copper cell. Macrophages in the bone marrow is known as a histocyte rather. In the spleen is called the follicular dendritic cell. The skin is called the Langerhans cell. In the kidney, we refer to them as mesenchymal cell. In the lymphatic system, we call them bile cells. And then in the lungs, we refer to them as alveolar macrophages. The entire process of production and maturation of granulocytes, usually from the stem cell stage down to the mature cells, usually takes a total of about 6 days. And this is very important to note to people. For this to occur, it requires the use of a number of growth factors that we'll be talking about in the next slide. The regulation of myelopoiesis involves a number of growth factors, and these growth factors include the granulocyte colony stimulating growth factor, the granulocyte macrophage colony stimulating factor, stem cell factors, and interleukin 3. To complete this model, let's talk about the different forms of pathology that could arise as a result of increased or decreased or abnormal production of white cells. Starting with an increased production of white blood cells tend to be referred to as leukocytosis. Decreased production tends to lead to leukopenia. Increased production of granulocytes in specific tend to lead to granulocytosis. If there's a decreased production of granulocytes, that we term that as agranulocytosis. Increased production of monocytes leads to monocytosis. Decreased production of monocytes tends to lead to monocytopenia. Wherever we have a neoplasia or malignancy of the immature precursor cells of granulocytes or the monocytes, like promyelocytes, the myelocytes, the metamyelocytes, and the band cell, usually we refer to that as acute myeloid leukemia. Whenever we have malignancies or mature granulocytes and monocytes, we refer to that as chronic leukemia. That concludes our module on myelopoiesis.